I'm Professor Jim Norman, and I'm a senior group leader from the Beatson Institute for Cancer Research in Glasgow, and my group focuses on the mechanisms of invasion and metastasis in cancer. The Beatson Institute is a core-funded Cancer Research UK institute, but we have a close association with the University of Glasgow. Within our institute building we have group leader positions that are funded by Cancer Research UK and also some positions that are funded by the University of Glasgow. The Beatson Institute is a basic scientific research institute that focuses on three major areas of cancer, three Achilles heels of cancer. We focus on cancer cell metabolism, we focus on cancer cell proliferation, and we focus on the mechanisms of invasion and metastasis, in particularly the contribution that cell motility and cell migration make to metastasis. This conference is part of a series of conferences that the Beatson Institute run. We run one main international cancer conference every year. This conference is focusing on cancer metabolism, really the role that energy metabolism plays in the progression of cancer. We have speakers that come from two fields of cancer metabolism, two parts of cancer metabolism research that are interlocked. The speakers that are at the conference, the keynote speaker is Professor David Sabatini from MIT in Cambridge, Massachusetts. But other speakers that we have is Mike Hall from Basel and Celeste Simon, also from the States. In 2015, the focus of the conference will be on cell polarity and the loss of cell polarity as cancers progress. One of the main focuses of our research at the Institute is invasion and metastasis, and how cell migration contributes to invasion and metastasis. AMSBIO produce a number of products which are specifically aimed at analysing cell migration within three-dimensional context, and those products are used widely within our institute.
I'm Professor Owen Sansom. I'm the Deputy Director at the Beatson, and I also run a research lab at the Beatson. What we're really trying to do is work out how the genes that go wrong in cancer might predispose to treatments. So certain mutations that occur very commonly will mean that cells will become resistant to certain treatments, but also sensitive to other treatments. And so what we try and do is find Achilles heels. So if a gene is mutated in 80% of colon cancer, like APC for example, what we hope is therefore we can find drugs that specifically target those cells that have that mutation. And I think that's probably our biggest contribution to drugs and cancer. Over the last 5 to 10 years, we've learned that cells don't just grow on their own. What the cells do is they grow in the context of signals that surround them. And one of the biggest things that surround cells is the matrix. And what you learn is that cancer cells are very dependent on the matrix that surrounds them. We use BME2 in culture. And so what this allows us to do is to take cells and cancer cells and grow them in an environment that is very much like in the body. And then when you put cells into that matrix, they'll grow and be very protected from many of the drugs that we try and hit them at with in vivo. So therefore, we're really important for us to use a sensible matrix that's like in the body. So therefore, when we're trying to work out how to kill cells, we learn how to kill them so that the surrounding matrix doesn't stop our drugs working. I'm really trying to work out how the matrix protects cancer cells from drugs. So I think the future of cancer is definitely personalised medicine, and I think our ability to work out the genes that go wrong in cancer and then be able to target the correct patient with the right drug will hopefully help us really add value to cancer research. And I think this is the big goal of many people working on personalised medicine as we go forward.
The Beatson Institute is a Cancer Research UK core-funded institute, and as such is part of Cancer Research UK's overall United Kingdom research strategy. Over the next 10 years, we have a programme in place to really focus on invasion and metastasis and cancer proliferation, taking our findings from basic observations that we make within the Institute into the clinic with our clinical collaborators within the west of Scotland and beyond.